Okay, so metabolism. You've probably seen this picture a million times in your other classes, so let me show it to you again. It's basically the network of metabolism and all the reactions that happen in your body. So you can consider metabolism as all the chemical and physical processes that break up or build molecules in your body and produce or use energy. Okay. Obviously, this picture here is very simple, is extremely simplified. Metabolism is actually much more complex, and there's many subcategories of metabolism that are not shown here. Maybe I'll show you this at the end. Okay. So I want to focus on this picture for one thing. When you read things online that tell you take this and it will change this, and take this and you'll do this, and take this and it will do this, from not so scientific sources, or at least some scientific sources but are doing poor research, you have to remember that you are reading a text that is talking about using one molecule, for example. So let's say they tell you take this supplement, and apparently that person that is telling you to take that supplement made sure that everything here does not inhibit what they're trying to achieve, or does not increase it, or does not have a side effect of it. Okay? So you have to think of it this way. If you add something in the network here, it will have an effect that will touch everything else. Okay? And because it touches everything else, it's extremely hard to predict what's going to happen to your body. Okay? There are computer software that are used by scientists, so we kind of do metabolomics, proteomics, lipidomics, using mass spec. And what this is in reality is that we extract proteins, lipids, and small molecules from cells. We put them in the mass spectrometer that you learned about in organic chemistry, and then it gives us a list of molecules, right, in your cell. The only problem with this is the metabolites, for example, if you extract them now or in 10 minutes, they will be totally different. Understand? So that's one thing. Second thing, your health state and your current diet and what you ate in the morning and whatnot affects this. And so if you're trying to study the effect of, for example, a supplement on your body, it's very complex to do. And so when you go and read online, people telling you do this and it will do that, and do this and it will do that, and they have nothing, it's not well researched, let's say, you have to be kind of cautious of these claims, okay? Because the person is basically telling you that they understand what it does to all this, okay? Obviously, to build a network like this, it took hundreds of years, okay? Not just like 10 minutes and that's it, right? Additionally, just from my own experience, so this is kind of a disclaimer for you guys, just from research, if you put 3 triplicates, because in research you're using triplicates most of the time, and then you extract whatever molecules there are in the cells, and then you run them, you'll have variation between them, even though you extracted them at the same time. And if you grow, you have mice, for example, and you're getting the liver of mice, and you're trying to study the content of the liver of the mice, and you're extracting the lipids and whatnot, and you run them, you'll find that the mice are different. Even though they grew together and they ate together and they come from the same mother and whatnot, it's highly variable. Okay, so it takes many years to kind of figure out what the hell something does to your body. Okay, right now we don't do it by hand. Before it used to be by hand. Now we just use computer software, and the computer software integrates everything and tells you the output. Okay, what else? Did I forget something? Additionally, when you extract stuff from a chemistry perspective, to extract everything in the cell, it's kind of complicated because everything reacts to different solvents and whatnot. So you'll have to choose different solvents, and by the time you do the extraction for at another time with a different solvent, you'll end up changing the whole thing. So it's very complex. Okay, that's the idea I want to tell you. In metabolism, there's 2 types. Okay, there's anabolism and catabolism, obviously, and these are important because your body needs to kind of take in stuff from food and then turn it in molecules that are relevant for the body. Because obviously if you're eating a plant, The proteins, the lipids, and the sugars in the plant don't necessarily apply to your body. You don't have, some of them you don't even have the enzymes to break down, right? And some of them you do. So, and the proteins for that plant, for example, most probably don't have a function in your body. That's what makes you different from the plant, right? But one thing that is common between them is, for example, the amino acids. And so you need to break down things and then rebuild them specifically for the body, okay? The process of building something is called anabolism. So you take simple molecules and you pack them together to build something that makes sense for the human body, okay? If you have a hard time figuring out which one is anabolism and which one is catabolism, you can remember this by just thinking about anabolic steroids, okay? So they're used by bodybuilders to gain mass. So you're trying to build something. Because me, I remember when I was studying this course, I had a hard time, even though it's kind of simple, but I had a hard time remembering the difference between them. I would get the exam and then suddenly I don't remember, okay? But if you want the trick, just remember anabolic steroids, bodybuilders, okay? So in anabolism, you're trying to grow, repair your body, and this requires energy. Anything that you're trying to build requires energy, okay? The energy, some of it is lost as heat, okay? This is important here. Catabolism is the opposite. You're trying to break stuff into smaller molecules. And you do this through, the first step is basically digestion, right? When you're starting to break down bigger molecules like the macronutrients into their smaller components. So proteins into amino acid, lipids into fatty acids, glycerol, cholesterol, and whatnot. Carbohydrates into the monosaccharides, okay? Additionally, catabolism is important for breaking up older cells. Older cells in your body are not good. They induce inflammation and that can cause problems. So you need to kind of get rid of them. So this is also catabolism. One feature or one characteristic of catabolism is that it releases energy, okay? And you'll see what I mean by this in a second. So when we're talking about adding glucose or when we're talking about generating or producing glycogen by using units of glucose, this is anabolism. It requires energy. And it makes sense, right? If you have too much glucose and you want to store it, that means you also have too much energy. And so anabolism will use energy and those units to produce glycogen. Then to break them down, that produces energy, okay? Same thing with triglycerides. Usually you take a glycerol molecule with 3 fatty acids, pack them together, this requires energy, and then to break them down will lead to release of energy. And proteins, make sense? Same thing. Okay, this energy, what is it? So the energy in your body is found in 2 forms, okay? You probably learned about the first one, which is ATP, but there's another one that is more long-term. This is creatine, and I'll tell you this in a second. It's not part of the slides, it's just for your own information. So ATP is an organic molecule that has an adenosine group and 3 phosphates bound to it, okay? Adenosine triphosphate. Each phosphate bond, when it's broken, it releases a pyrophosphate, just the P with a small I, and releases energy. And this energy is important for reactions that are of the anabolism category, let's say, okay? When you eat, let's say, for example, too much sugar, sugar is going to be used to produce ATP, and so this will start with the AMP, adenosine monophosphate. You'll have a phosphate group added to it to produce adenosine diphosphate, and then another pyrophosphate added to it to produce ATP, okay? Now, ATP's lifespan in your cells is very short. So you need another form to keep that energy. So there's another cycle called the creatine cycle, and this creatine cycle will take, this is just for your information if you're thinking about working out or whatnot and taking creatine. Creatine will steal the energy from ATP and become phosphocreatine. and phosphocreatine's lifespan is much longer. Okay, and so if you're, for example, going to the gym and you're trying to work out and you take creatine 30 minutes before, what your body is going to do is that it's going to load that creatine with energy and keep it in the form of phosphocreatine. So when you start working out, when your body needs to push more than what you can do usually, it will break down that phosphocreatine, okay, to release the energy. Make sense? This is why people usually take creatine before, about 30 minutes before they start working out, okay? You can also take it after because it has some regeneration features, but that's out of the scope of metabolism, okay? Cool. Okay, where's this ATP produced? Most of it, but not all of it, is produced in the mitochondria. One important thing that I want to talk about the mitochondria is that you have the matrix, where I'll get back to this in a second, but the matrix is where the TCA cycle happens, and we'll talk about the TCA cycle in a second, while the intermembrane space is where the oxidative phosphorylation happens. And these are two processes that will produce energy. Simple? You've seen this before many times. Okay, so what are the three chemical reactions that are fundamental to metabolism, right? So there's three major classes. You have condensation and hydrolysis. Obviously, one of them is for anabolism, the other one is for catabolism. You have phosphorylation, dephosphorylation, and you have oxidation and reduction, okay? And we'll take a look at these three one by one. So, many reactions in your body just require water, okay? Nothing crazy. Actually, most of them require water. So, condensation is when you're trying to build more complex molecules, okay? So, for example, when you're trying to combine glucose with fructose to produce sucrose, this is a condensation reaction. Why it's called condensation? Because you're losing water in the process. Hydrolysis, you're using water to break up a molecule, okay? So this is anabolism, and this is catabolism. Simple? Okay. Next. Phosphorylation, dephosphorylation. This one I just explained with ATP, but phosphorylation is basically the addition of a phosphate group to a compound. For example, the glycerol backbone of a phospholipid will get phosphorylated, right? Because you want to add a phosphate group to the glycerol. So this is a phosphorylation step. Dephosphorylation is the removal of a phosphate group. And obviously, if you're removing a phosphate group, then you are releasing energy. If you're adding a phosphate group, then this requires energy. Okay? Make sense? For instance, this is a good example. Glucose can do a lot of things. It can be used and oxidized to produce energy. It can be stored as glycogen, right? So glucose gets phosphorylated, and this phosphorylation allows it to go through the glycolysis pathway and then to enter TCA cycle and whatnot, okay? Movement of the phosphate group from one carbon to the other leads to it going to produce glycogen, okay? So if you just ate, it will go like this, and then when you have too much energy, the remainder will go to glycogen. Additionally, you can dephosphorylate glucose, and if you remove the phosphate group, right, then you go back to your glucose with a pyrophosphate. Make sense? Okay, this is where students might get lost. The third type of reactions that happen in your body are oxidation and reduction. If you want to remember this easily, because you've probably seen it in the past, a molecule donating an electron is oxidized. Okay, this one is a bit hard because it doesn't make sense, but a molecule acquiring an electron is reduced. You can remember it this way. It's reduced because its charge is decreasing, so it's becoming more negative. Okay, make sense? if you remember from your biochemistry class, you had to think of who's the reducing agent and oxidizing agent and whatnot. In this case, I'm not asking you to remember that. There's no purpose, okay? At least for nutrition. So, is this clear? Is this trick simple? Whatever gets reduced is whatever is gaining an electron. It's becoming more negative, okay? So for example, FAD plus, there's a plus missing here. It has a plus here, now it does not have a plus here anymore, so its charge is being reduced, okay? It goes from positive to neutral, okay? So it's being reduced. Simple. Obviously, this question here is just referring to the rest of the lecture. I'll show you this in a little bit, but you've probably seen it before, the TCA cycle, glycolysis, and the oxphos. Okay, so when we're talking about metabolism, we're talking about reactions, basically. One molecule converted to the other, until you reach the product, right? And so this happens through enzymes. These enzymes are the one that catalyze the chemical reaction, but they don't do this work alone. They do this work with cofactors and coenzymes. Cofactors are non-proteins that enhance the enzyme activity or are necessary depending on what we're talking about, okay? Some of them, if you don't have the coenzyme, then the reaction does not happen. For example, FAD, FADH2, and vitamins work as coenzymes. This is why vitamins are very important, and deficiency can cause a lot of problems. It's because they act as coenzymes for a lot of the reactions that happen in your body, okay? Cofactors, on the other hand, are typically minerals, so iron, magnesium, zinc, and they're required for enzyme activity. For example, hemoglobin has a cofactor, which is iron, and this iron is important for oxygen uptake. Okay? This is just a diagram showing you what I'm talking about. So the enzyme, in this case, requires its coenzyme to prepare kind of the enzyme to bind to the compound. Because if that coenzyme is not there, then this space here is not going to take up the compound, okay, and cannot metabolize it. So this is just a simple diagram of how this could work. Simple? Okay. So now the complicated stuff with a lot of numbers. How is energy extracted from carbohydrates? So we'll look at carbohydrates, then we'll look at lipids, then we'll look at proteins, and then we'll look at alcohol, and that should be it for the lecture, okay? So for carbohydrates, they are broken down into smaller units. They're broken down into glucose, fructose, and galactose. Glucose is transported in the liver, it gets phosphorylated, and either stored as energy, as glycogen, or released for other cells, okay? When it gets to those cells, then it undergoes glycolysis, TCA cycle, and whatnot, okay? The excess glucose gets converted into fatty acids and glycogen, okay? Make sense? And these fatty acids are then stored as triglycerides in your adipose tissue. Fructose and galactose are also converted to glucose. So everything goes to glucose. Galactose is converted through a pathway that we're not going to talk about. It's called Leloir pathway, which takes galactose, and if you remember, just quickly, you probably know it, it's adding UDP to it, and then just goes on a few reactions and becomes glucose. Fructose also has its own pathway to be converted to glucose, okay? And then this glucose gets oxidized through glycolysis, TCA cycle, and oxphos, oxidative phosphorylation. Glycolysis happens in the cytosol of the cell. That's basically the liquid that's inside the cell. It is an anaerobic reaction. It does not require oxygen. Okay, and what you need to remember about this is that a 6-carbon glucose gets broken down into 2 3-carbon molecules, pyruvate, which can be used to then synthesize, either go inside the TCA cycle, or it can be converted back into glucose through gluconeogenesis. Okay, so this is where gluconeogenesis comes in. One important thing to remember is that ATP is both used and produced in this process, and the net number of ATP produced is 2. Okay, so 2 of them are used, 4 of them are produced, and so you end up with 2 ATPs. Okay. So this is an anaerobic process in itself. Now, after this happens, if you don't have oxygen, pyruvate is converted into lactic acid. If you have oxygen, then it will be going inside the TCA cycle. Okay. In this case, NADH is going to be oxidized, so it becomes more positive to NAD+, and these are just proton carriers that are important for what we're going to talk about in a few seconds. Okay, make sense? So if I tell you on the exam there's no oxygen, how much ATP are you producing from glucose? It's going to be 2. Okay. 1 glucose molecule, 2 ATP under no oxygen. Questions? Simple. Good. Okay. Something interesting. So pyruvate, when there's no oxygen, it will be converted to lactate. Okay. And it will require the oxidation of NADH. Okay. Now, in your muscle, when you are doing anaerobic exercise and when you're kind of, let's say for example, sprinting, okay? When you are sprinting, this is a hypoxic exercise. It means that you're not taking up as much oxygen as you should be, and so glucose will be converted into lactate, okay? AKA lactic acid. There's a myth that says that the soreness after working out, for example, in your muscles is due to lactic acid, okay? That's not true. Do you guys know where it comes from, the soreness? No? Anybody? It comes from just the pH of your muscle decreasing. Okay, this is just acidosis. It's, how many of you believe that it was lactic acid? A few, right? Yeah. Well, that's not true actually. That's some stuff you, yeah. No, actually it's due to this process here, okay? It does make sense, indeed, but lactic acid gets converted in acetic acid also, and so it's not the lactic acid itself. It's just because this is the molecule that was accumulating in the muscle, and so people just said, well, it must be it, right? But it's not. This lactic acid or lactate is then transported to the liver, and lactate can be converted back to glucose. Okay, make sense? Okay, if you have oxygen, so this is where it gets complicated. If you have oxygen, then the pyruvate does not become lactate. Instead, it will become acetyl. It will make 2 molecules of acetyl-CoA. It will get activated basically by coenzyme A here. And this CoA will just bind to the pyruvate, and there will be a release of CO2, 2 CO2 molecules. Okay. I am not expecting you to remember the reactions themselves. Okay. What I say by the reactions, I'm not expecting you to know each step, how many NAD+ are used to produce NADH and how many CO2 are released and whatnot, but what I expect you to know is the general effect of the whole thing. So for example, glycolysis, under no oxygen, you need to know it's 2 ATP. With oxygen, you need to know that it's between 30 and 38. Okay, and I'm going to show you this in a second. So these numbers are important. The rest, what happens at which step, this is not for this course. Okay. So acetyl-CoA is a very important molecule. It's what everything gets converted into. right? This is why you have the ability to produce energy from amino acids, for example. This is why you have the ability to produce energy from fatty acids. It's because everything, even alcohol, gives you energy, right? Technically, not the next day, but technically. So everything gets converted into acetyl-CoA, and this acetyl-CoA will enter the TCA cycle and undergo oxidation, and then in the mitochondria will also contribute to oxphos. Okay? I'll come back to this, but amino acids can be differentiated into two categories. Glucogenic amino acids will enter at the pyruvate, so in the glycolysis pathway. Ketogenic amino acids can produce fatty acids, and so they will enter at acetyl-CoA. And I will show you how this is happening, okay? So ketogenic amino acids are certain amino acids from the 20 that you learned that are going to be transformed into fatty acids if they are in excess. Glucogenic amino acids are going to be giving you energy if you are lacking energy from your food, okay? TCA cycle. So the TCA cycle is where you have the two acetyl-CoA molecules that will enter this cycle of reactions, each using NADH, NAD plus producing NADH, or FAD plus producing FADH2 to produce energy, okay? So the TCA cycle has 8 reactions. It happens in the matrix of the mitochondria, that's inside the mitochondria completely, not in the intermembrane space. And for each acetyl-CoA that enters the TCA cycle, you will produce 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of ATP, and 2 molecules of CO2. Okay? GTP, sorry. This is a mistake here. Okay? GTP. Actually, let me send an email for this, okay? I just want to verify this because I think there's a mistake here. These 2 make sense, these 3 make sense, just the ATP stuff I want to check. Okay? Yeah.
But GTP can be converted into ATP.
Yeah.
So in the end, it's kind of the same thing, or it's just from the actual TCA cycle?
GTP does not get converted into ATP. It's just the transfer of the phosphate that goes from one to the other. But let me just clarify this because it's not clear here, okay? So for now, just forget about that slide. Or that part of the slide at least. Okay, oxphos, the last step. So why are you making FADH2 and NADH? It's because they're necessary for the for oxphos, okay? And so this takes place into the inner membrane of the mitochondria, where you have kind of an electron transport chain, which are basically a few transporters on the membrane that will carry, that will basically take up an electron and then carry it through the chain. And while this is happening, it opens up the pores that release the protons outside or bring them back inside. And this changes the proton gradient along the membrane. And this is going to be important to produce energy, okay? All this stuff here is from biochemistry. You don't need to know it for our purpose. I'm just explaining why do you need NADH and FADH2, okay? They basically will provide the electrons for the electron transport chain, okay? Each enzyme in there, in the electron transport chain, will convert NADH in NAD plus and FADH2 in FAD plus, okay? And oxygen, this is very important. This is why you need oxygen. It's the final acceptor of electrons, okay? So you've probably never seen oxphos like this because we don't really care about what happens in there, okay? My only thing that I want you to remember is that one NADH molecule gives you 3 ATP, one FADH2 gives you 2 ATP, and the complete oxidation of one glucose gives you between 30 to 38 ATP. Do you guys know why? Yeah. 32. Exactly. It depends on a lot of things. So I'm going to ask you this actually. Why is it a range and not a specific number? And why can't we get the number? Yep. Go ahead. Sorry?
Depends on the molecules that are used.
Well, if we're talking only about glucose. Yeah?
Now you just confused me, but I was going to say that depending when the cycle starts, because some intermediates come from different sources.
Yeah, that makes sense if we're talking about the whole metabolism. But if we're talking about one molecule of glucose only, let's imagine you remove everything else. Okay, I'll tell you. So the reason why we cannot get this number, and it was the same thing when I was studying, they don't seem to be able to get the number. In every biology book I read or biochemistry, they have a different number, okay? The reason for this is that the electron transport chain is leaky. And because it's leaky, sometimes the number of protons that are going through, it's more than the reaction before, okay? So it's an inefficient process, let's say. Because it is an inefficient process, sometimes it reaches the maximum amount of ATPs produced, and sometimes it does not, okay? But if we're talking about metabolism in general, then yes, it makes sense. NADH and FADH2, they come from different places, okay? Depending what molecules you have, then you will either be making NADH or FADH2, and this is what each one gives you, okay? But just for your information. Okay, so fats. How do we make energy from fats? Fat metabolism, I'll tell you now, the end goal of it is to produce acetyl-CoA. It's always to produce acetyl-CoA. It's the molecule that you need to then do whatever you have to do. And it's the molecule that is at the center of all energy metabolism, okay? So fat metabolism, the way it goes is that you need to break up the bigger molecules that you have, for example, triacylglycerides or cholesterol esters and whatnot. And this is the process called lipolysis or lipolysis, depending on how you pronounce it. It will break two things. It breaks the dietary fat in your food and the adipose fat that you have in your body, okay? And this is broken down by an enzyme called lipase. Obviously, this lipase here is just a general term. There are many different types of lipases, and so depending on which molecule you're dealing with, it will be a different lipase, okay? So this is just a category of enzymes that break down lipids. This will produce a glycerol molecule and 3 free fatty acids when you're breaking down a triacylglyceride. This is the storage form of lipids in your body, okay? Glycerol can be converted into pyruvate because they're both 3 carbon molecules, okay? And then this pyruvate can be converted into acetyl-CoA the same way it is converted in post-glycolysis, okay? Fatty acids are broken down into pieces that can then be used to make acetyl-CoA, okay? And these can be used for energy as well. So, in other words, you have your triacylglycerides, there is a lipase that will break them down into 2 pieces. Glycerol will go to the liver, produce pyruvate, and then pyruvate can then make acetyl-CoA or can make glucose, okay? Depending on which direction you're going in metabolism. If you need energy, you'll be making acetyl-CoA. If you have too much energy, you'll be making glucose, okay? The fatty acids, they're released in the bloodstream, as we've seen in the digestion class and in the lipids class, and they will be carried to be broken down somewhere else or stored again. So, when fatty acids are produced, the short and medium chain fatty acids are attached to albumin. This you have it in the diagram where you basically have the kilomicron being made, and then it goes into the lacteal. While next to it, you have that line that shows you the short and medium fatty acid chains that are attached to albumin, okay? And this helps it be transported in the blood, okay? For long chain fatty acids, they are first activated by a coenzyme A, so CoA. into fatty acyl-CoA, and that is going to be taken inside the mitochondria by a shuttling protein called carnitine. Okay? If you've ever tried to lose weight and you've looked online, you've probably seen people telling you to supplement with carnitine or acetylcarnitine. So this is a shuttler, and the idea behind it is that carnitine will act as a transporter, and because you have more of it, it will take up more lipid and bring them in your mitochondria to be broken down. Okay, not so simple. Okay, there's little shown that it actually works. Okay, then when the fatty acyl CoA, so fatty acyl CoA is just a long chain fatty acid with a CoA, enters the mitochondria, it undergoes beta oxidation, which is to break it into 2 carbon segments to form, and then activated with a coenzyme A to form acetyl-CoA. Okay. In other words, imagine a long chain of fatty acid. It goes into the mitochondria and gets broken down into 2 carbon molecules, right? Because the side chain of the fatty acid has, let's say, 16 carbons. So it gets broken down into 2 carbon molecules and then gets activated with a CoA addition. Okay? And this, if you remember, is the center of all energy metabolism. Okay, back to this. Let's take an example, for example, palmitate, which has 16 carbons. So this 16 carbon palmitate will be broken down into 2 pieces at a time. The CoA will be added to it. And one of the role of the FAD being turned into FADH2 and NAD plus being turned into NADH is that it's going to pick up the extra protons that are going to be released from breaking this carbon. If you remember, each carbon has hydrogen atoms on it. So these hydrogen atoms need to go somewhere, right? And so they will be basically used for these reactions. And then you can do this many times. So for a 16 carbon molecule, if you undergo full beta oxidation, you'll produce 8 acetyl-CoA molecules, 8 FADH2, and then 8 NADH. Okay, simple. What else? This is not 8 NADH and 1 H plus, it's 8 NADH and 8 H plus, okay? Simple. So this is why fatty acids produce a lot of energy. Okay, this is why when you're counting, for example, how many grams, how many kilocalories you're producing from fat, and you produce 9, it's because of this process here. Okay, this is just the same way to show you this. In general, so you have your fatty acids that undergo beta oxidation. Everything meets up right here at acetyl-CoA, and then you have the same stuff that happens for the rest of the way, which is TCA cycle and the electron transport chain. I gave you the answer. Enough questions for today, but why do they produce more energy? Well, it's because of the chain. So yeah, so it's this here. This is not true, okay? They cannot be converted into 4 molecules of pyruvate. You have a 3 carbon molecule converted to a 3 carbon molecule, you cannot make 4 out of it, out of 1, you know? So the answer is B. If you take a look at their structure, you can see that they're almost similar, okay? The only difference, or the main difference, is that one is longer than the other, and this is why it produces more energy. There are more bonds to break. Okay. Okay. Juan ate a well-balanced meal 3 hours ago and is resting on the couch. Which metabolic pathway is providing most of his ATP? Three hours ago. That's a key. Yeah, yeah, because you are relaxing, okay? You're laying down on the couch. So when you're laying down in the couch and you're in between meals, it's your lipids that are being used, not the glucose. Glucose usually is short term when you need it quickly, but then after maybe like 30 minutes or so, it reverts to, obviously it depends how much sugar you ate there. But if you remember that figure that I showed you where you're eating too much sugar and then it goes down quickly. So if you eat too much sugar, it goes up and goes down in like 1 hour. Obviously, if your meal has low levels of sugar, then you remember that curve there that goes kind of slowly? That takes about 2 hours. But as I told you, if you are relaxing, if you are relaxing, then it's going to be lipids between meals. Yes?
I don't know his explanation. He's not doing anything, he's just laying on the couch, so he's a beta. Laying on the couch, he's oxidizing.
That's it. That's a good way to remember it. You should be doing stuff in life, not laying down on the couch, you know. Gotta be enough, I guess. Yeah. Anyways, so question number 3. Which intermediate is common to catabolism of fat and glucose? Anybody? Good morning, guys. Yeah, it's acetyl-CoA. Okay, proteins. Proteins. So you can also produce energy from proteins, but the body prefers fat and carbohydrates because proteins have a lot of other functions that cannot be performed by the other compounds. Okay, protein is only used for fuel if your total energy is kind of low or your carbohydrate intake is low. Okay. And so the way to break down proteins to produce energy is through the process of proteolysis. And this is just basically digesting the bigger proteins into smaller amino acids, into peptides, and then amino acids. And then these amino acids are transported to the liver, where they can be either remade into bigger proteins or released in the blood for other functions. Okay, the excess protein or the excess amino acids that result from the proteins are going to be used for energy or stored as triacylglycerides. Okay, this is why when you're working out and you're taking too much proteins over the recommended amounts, it actually has the opposite effect of muscle building. It can end up being stored as fat. Okay, so there's a sweet spot that you have to kind of figure out. Too much of it will lead to actually making triacylglycerides, and if you're trying to build muscle, well, you will build the muscle, but at the same time there will be fat covering. Okay, that will build up. Okay, how do you make energy from amino acids? So there's a process called deamination. And if you remember, I talked about transamination, where you're transferring an amine group to a keto acid. Okay, deamination is removing the amine group, which produces a keto acid. The keto acid is very similar to acetyl-CoA. Okay, right? And because it is very similar to acetyl-CoA, then it can be used to produce acetyl-CoA, which then enters the TCA pathway. Okay, make sense? So, is this clear, the deamination? So, when you're undergoing deamination, one of the side effects of deamination is the production of ammonia. And ammonia is not good for you, so it needs to be kind of taken care of. And the way it's being taken care of is by producing the urea that we talked about, and then get excreted. So urea is less toxic than ammonia. This is why when, for example, people don't eat or don't take up enough energy, and then their body has, for example, during starvation or famine, and then the body has to kind of use its own tissues and break them down to produce energy, then it will produce a lot of ammonia because it's not clean energy, let's say. And this can cause a lot of problems. Okay? The keto acid is the carbon skeleton that will be channeled into glycolysis or the TCA pathway. Now if we go back Just 1 second, I should put these slides. This is what I mean, okay? So you have certain amino acids that are glucogenic that will be deaminated. When they get deaminated, they will produce a keto acid that will be converted into pyruvate because their structure resembles each other. If you have a ketogenic amino acid, it will be deaminated and it will produce a keto acid that resembles acetyl-CoA, and that will be converted into acetyl-CoA. Just 1 second. And this is pretty much it. Okay. Yeah. What else? Okay. So there you go. If you needed some examples of which ones are glucogenic and which ones are ketogenic, this is it. You don't need to know them by heart though. Okay. Right. So the ammonia that's left can then have two fates. Either it can be used to aminate, let's say, it can be added to other keto acids to produce other non-essential amino acids. The remainder of the ammonia is converted into urea, and if you have too much ammonia, that is toxic for you. Okay. I think there's a plus missing here. Okay, so what are the end products of deamination? Buddy? B? Yeah, ammonia and the carbon skeleton. Alcohol. So even alcohol can produce energy, okay? So how does it get metabolized? So alcohol is primarily oxidized or metabolized by the liver. The first enzyme that takes care of this process is alcohol dehydrogenase, which converts alcohol into acetylaldehyde. Acetylaldehyde then gets converted into acetate or acetic acid, which is an acid, obviously. And if this gets released in your blood, it decreases the pH of your blood. So this is the one of the dangerous parts about alcohol, okay? There's another system called the microsomal ethanol oxidizing system, which is an alternate pathway that can oxidize alcohol. And this happens in all cells in the microsomes, okay? But it does not get activated unless you are chronically, unless you chronically consume alcohol or you actually abuse it, okay? Usually these are sufficient. Sometimes there's a another system that gets activated to take care of this, okay? The MEOS. Okay. So alcohol is a 2-carbon molecule. So actually all the alcohol that you drink, the thing that does whatever it does, the effect in your body is ethanol, okay? So any alcohol has ethanol in it. Methanol, which is similar to ethanol, is actually toxic for you, okay? So methanol does not get you drunk, it gets you dead. Ethanol kills you slowly, right? So this is the, when you're reading 37% and whatnot on drinks, it's the ethanol, okay? The ethanol content. So ethanol, through the ADH enzyme, will produce acetylaldehyde, so alcohol dehydrogenase, and then acetylaldehyde will be converted into acetate or acetic acid through the acetylaldehyde dehydrogenase, and acetate can be then converted in acetyl-CoA. Okay? Make sense? So, I told you that acetyl-CoA is at the center of everything. So, if it's at the center of everything and you're consuming too much alcohol, what happens to the acetyl-CoA? Yeah, exactly. So it gets converted into fat. And so this is why a lot of people that abuse alcohol, usually they're overweight, okay? Usually. Yeah?
Doesn't it also fatty liver?
Yeah.
Or- okay.
Yeah, I'm 3 slides down, or a few slides. Yeah. So there's also a little bit of oxidation of alcohol that happens in your stomach. Okay, so the first place where alcohol gets actually converted is in your stomach, but most of it happens outside of the stomach. It happens in the liver. Okay, so in the stomach, there's this gastric alcohol dehydrogenase, which will start oxidizing the alcohol. Okay, and this will signal to reduce alcohol absorption. Okay, because you don't want to absorb it too fast. The amount of this enzyme in your body is different from people. There are genetic differences linked to this. This is why some people get drunk faster than others. Okay, some of them have a lower effect of reducing alcohol absorption, and so it goes in their bloodstream much quickly, and because it does so, it has an effect much more quickly. Okay, if you reduce the alcohol absorption, for example, you have a mutation that makes it even stronger, then you get drunk more slowly. Okay, so this is part of it. Most of it is absorbed in the intestine, goes in the blood, same way that you learned for any other nutrient or molecule, and then goes to the liver. Some of it is in the stomach, as I said. Most of it is in the intestine. And the liver typically oxidizes alcohol at a 1 drink per hour rate. Now, obviously, each drink is different in size, but there's a specific amount in terms of milliliters of pure ethanol, 17 milliliters, something like this. Okay, this is what's considered the drink. The rate at which you are oxidizing this alcohol depends on many things, including your profile, your health, how much of it you took the last week, your body, etc. Okay? And then the excess alcohol goes back in the blood, and this is not good, okay? Because it can react with many things in your blood and become toxic. For example, one of the side effects of alcohol accumulation is the production of acetaldehyde, and this inhibits protein synthesis, increases free radicals, and increases lipid peroxidation. So this is the free radical. When a radical attacks the lipid, it creates a lipid peroxide, and these are dangerous molecules for your body because they can basically destroy membranes of your cells. Okay? When this happens, the MEOS pathway has to become more active to take care of whatever you have extra alcohol that you need to metabolize. And the MEOS has other functions, but they will start prioritizing alcohol metabolism to kind of decrease the amount of alcohol you have in your blood. Okay. So this is the amount of what I was talking about, which is in pure ethanol. Okay. It's not 17 milliliters of vodka or whatever. This is the pure amount in it. Did I say anything else? Yeah, it's similar to carbohydrates. Okay, this might be a little bit interesting. If I'll ask the question, probably no one will answer. Anybody not remember a night? Nobody want to answer? Okay. Well, if you answered in your head that you did forget a night, I'll tell you where you were. You were here, in this range. Okay? This is your blood alcohol concentration. The legal limit for driving is 0.08. If you have a learner's permit, it's 0. Okay? If you have the full permit, it's 0.08. At this point, you're still able to drive and function properly, but starting to be a bit weird. At this level here, you start not making memories. So your memories just stop being made. So if you've blacked out, that means you killed it that night. Be careful, okay? Because the next level, and at this point, you basically lose the judgment. So it's very easy to get to this point here, okay? Because you still make sense, but first, your body is not making the memories, but the other thing is that this is where usually you start vomiting also. So usually they happen together. If you vomit, you probably blacked out also. The problem with this place here is that your judgment is not there anymore, and so it's very easy to get here, and this can kill you. Okay, so just a little bit here, and you're off. Okay, so if you drink, try to stay here. Okay, why is it important for nutrition? Well, for this. Sorry guys for this if you don't like this kind of pictures. Your liver usually looks like this. Okay, as you drink alcohol, what happens is that if you abuse alcohol and you drink too much alcohol, it will be going to your liver. And if it goes to your liver, the excess will be making lipids. And if you're making lipids, it will overload the liver. So when it overloads the liver, you end up having a liver with a fat layer on top of it. Okay? Fat on top of the liver ends up leading to inflammation. Okay? And when you have inflammation, it can cause a million different things. One of them is the destruction of your liver. Okay? And usually when you get to this point, cancer starts appearing, and you have many problems that come with it. Okay? There's a place in between these two phenotypes, or these two livers, where you can revert back. It's when you abuse alcohol too much and you have a fatty liver, but you start working out and giving time for your body to kind of burn whatever fat is on top of the liver. Okay? Make sense? Good. But usually this happens much later on in life, around the 40s-ish, and that's if you abuse alcohol constantly. I worked on a similar disease as this, but it's kind of the equivalent of it without the alcohol, so it's called non-alcoholic fatty liver disease. So this was part of my postdoctoral work. Okay. Right. So this slide, what you need to take out of it is that energy, the only thing that has no storage mechanism is amino acids or nitrogen. So for fatty acids, for example, you can store them as fat into triacylglycerides into your adipose tissue. Carbohydrates can be turned into glycogen, which is the store of sugar in your liver and muscle cells. But amino acids and nitrogen don't have any. So this is why they get excreted more often. Okay? And the stored energy is going to be used at times where you're not eating or where you're consuming too much energy. So that means during sleep, fasting, or during exercise. Okay?